Dividing phase of the cell cycle, cell division. Cells, they divide by two types of divisions. So there are two types of cell divisions occurring in the cells of living organisms. The first one is mitosis and second is meiosis. Mitosis is a cell division in which one parent cell divides into two daughter cells with the same number of chromosomes as chromosomes present in the parent cell. This type of cell division occur in body cells. Body cells means somatic cells. Whereas in meiosis, one parent cell divides into four daughter cells with half the number of chromosomes that are present in the parent cell. This type of cell division, it occurs in the sex cells, means the cells of the reproductive organs, to form gametes. Now starting with the explanation of mitosis. Beta, mitosis. Mito means thread. Thread means chromatin thread, that is chromatin fiber. So mitosis is a cell division in which one parent cell divides into two identical daughter cells. That is the most important point of mitosis, that here in mitosis, one parent cell divides into two identical daughter cells. And the second point related to mitosis is the same normal chromosome number is maintained at each cell division. Means after the cell division, as one parent cell divides into two daughter cells, those two daughter cells have the same number of chromosomes as in parent cell. Now this process of mitosis is divided into two main phases. The first is karyokinesis and second is cytokinesis. Beta, karyokinesis. Karyo means nucleus and kinesis means division. So karyokinesis is a process in which division of nucleus takes place. And further, it is divided into four phases, four continuous phases: prophase, metaphase, Anaphase and Telophase. All these four phases occur in such a way that these phases merge into the next phase, making it a continuous process. So the division of nucleus in cell occur in these four phases: prophase, metaphase, anaphase and telophase, which we are going to study in detail. And after the division of nucleus, the second phase of mitosis is cytokinesis. Cyto means cytoplasm and kinesis means division. So in cytokinesis, division of cytoplasm take place, which divide the parent cell into two daughter cells. So let's begin with the four phases of karyokinesis in mitosis and afterwards with cytokinesis also. Now, in interphase, that is the resting phase of the cell cycle, cell prepare itself for the cell division. So if cell enter the synthesis phase of interphase, then it prepare itself for the cell division, and at the end of the interphase, cells looks like this. Beta, mitosis occur in all type of body cells, means it occurs in animal cell also and in plant cell also. Only a little bit differences are there, which we are going to study side by side when we will study about different phases of mitosis. Now, at the end of the interphase, animal cells looks like this. Labeling of the diagram, labeling of each phase is very important. So, in animal cell, this is the plasma membrane. So, this is cell membrane or plasma membrane. Here cytoplasm is present. Now this is nuclear membrane. Inside the nuclear membrane, these long thread-like structures, they are chromatin fibers. And this spherical structure, it is known as nucleolus. Now, nuclear membrane, chromatin fiber, nucleolus, as it is an animal cell, just very close to the nucleus, this a small spherical cell organelle. Centrosome, it also take part in cell division in animal cell. Now, plant cell wall, the outer layer of plant cell is cell wall. Again the same nuclear membrane, chromatin fiber, nucleolus. No centrosome is present here. So like these cells, at the end of the interface, animal cell and plant cell looks like this. So as they are ready for the cell division, so they enter first phase of dividing phase, that is mitosis, prophase. But now in prophase, the chromosomes, the first thing which appear, now these are the chromatin fiber. So as the cell prepare itself for cell division, so these chromatin fiber, they have become short and thick and clearly visible inside the nucleus. So that is the first change which occur at the beginning of prophase, that chromatin fiber, they get condensed, and due to which they become short and thick and they are clearly visible inside the nucleus. So 3 chromosomes I am drawing here. So chromatin fiber, they become short and thick and clearly visible inside the nucleus. In the same way, in plant cell also, the chromatin fiber condense to form short and thick, clearly visible chromosome inside the nucleus. Now, each chromosome has already duplicated, means having 2 sister chromatid in it, which are attached to each other at a point of attachment called as centromere. So here also chromosomes, they are duplicated and their sister chromatids, they are attached to each other at a point called as centromere. Now after this, the next change which occur in animal cell, the centrosome splits into 2 along with simultaneous duplication of the centrioles contained in it. The centrosome, which is present near the nucleus of the animal cell, it splits into 2 parts, and the 2 centrioles which are present inside the centrosome, this, as soon as it splits into 2 centrioles, these 2 centrioles, they start moving apart from each other towards the imaginary poles of the animal cell. So this is the first change which occur in prophase in animal cell, that centrosome splits into 2 centrioles. Both the centrioles, as soon as they divide, they start moving apart from each other towards the imaginary pole of the cell. As they move, in between them, spindle fiber formation takes place. At the end of the prophase, these centrioles, they have reached the imaginary pole where they are surrounded by radiating rays known as asters. So asters are the radiating rays which surround the centrioles on reaching the poles of the animal cell. And spindle formation, the fibers, long thread-like fibers, they are formed in between these 2 centrioles. these spindle fibers, they help in holding the chromosome in their position during cell division. So this is also the one of the change which occur in prophase. So the first change, that is division of centrosome. As centrosome divide, centrioles start moving apart from each other. As they move apart, spindle formation takes place in between two centrioles. Now, as soon as these centrioles reaches the pole of the animal cell, there they are surrounded by radiating rays called as esters. After this, the chromosomes which are present, they start moving towards the imaginary center or the equator of the cell. So these chromosomes, they start moving towards the center of the cell. And the last change which occur at the end of the prophase, this nuclear membrane, it start disappearing. The nuclear membrane and the nucleus which is present here inside the nucleus, both disappear at the end of the prophase. Now the chromosomes has to move out of the nucleus for division. So that's why this nuclear membrane, it disappear. Along with nuclear membrane, nucleus also disappear. Now in plant cell, as no centrosome is present in plant cell, so there spindle formation takes place in plant cell also, but in plant cell, this spindle formation takes place due to condensation of microtubules present in the cytoplasm. As centrosome is not present here, so the formation of spindle fiber in plant cell takes place due to condensation of microtubules present in cytoplasm. Microtubules are the type of proteins which condense to form spindle fiber in plant cell. So spindle formation takes place in plant cell during prophase. Chromosome, as in animal cell, they start moving towards the center of the cell in plant cell also, and the nuclear membrane and nucleus disappear in plant cell also. So these are the changes which takes place during prophase in plant and animal cell. Let's revise these changes as given in your book. Just see the diagram. This is prophase. In your book, the term is written early prophase and late prophase. So the changes which takes place at the beginning of prophase, that is early prophase, and the changes which takes place at the end of the prophase, that is late prophase. But while identifying the stages in diagram, you have to write prophase only. Don't write early or late with it. You have to identify it with the main name, that is by the name only. Don't use these prefix with it. Now just see the first change in animal cell. Centrioles start moving apart from each other to reach the opposite pole. Now second, chromosome, they are clearly visible inside the nucleus. They are duplicated, having two sister chromatids attached to each other at centromere. Now at the end, these chromosomes, they start moving towards the center. This nuclear membrane and nucleus, it disappear at the end of the prophase. Centrosome divide into two centrioles. These centrioles move apart from each other towards the pole, where they are surrounded by these radiating rays called as esters. In plant cell, as centrosome is not there, so the spindle formation takes place by the condensation of microtubules, and here also the chromosome at the end of prophase, they start moving towards the center, and nucleus and nuclear membrane disappear. Now the next phase, after this, cell enter next phase of the mitosis, that is metaphase. So during metaphase, only two changes takes place in animal and plant cell. Now what are those two changes? Already the nuclear membrane is disappeared. Chromosomes at the end of the prophase, they had start moving towards the center. So in metaphase, chromosome line up at the center of the cell, that is at the one plane of the equator. So at the center of the cell or at the equator, they line themselves at the center of the cell, and each chromosome, they get attached to the spindle fiber by their centromere. So these are centromere. So spindle fibers which were already formed in between centrioles, so those spindle fiber, they get attached to the centromere of each chromosome to hold them in position. So these are the two changes. So the chromosomes, they align themselves at the center of the cell, and the second change, that the spindle fiber, they get attached to the centromere of the chromosomes. Like in animal cell, in plant cell also the same changes occur. Chromosome align themselves at the equator, that is the center of the cell, and spindle fiber, they get attached to the chromosome at the centromere. So these are the spindle fiber. So these are the two changes. After metaphase, now the cell enter next phase, that is the smallest phase of the mitosis, and we call it as Anaphase. Longest phase is prophase and the smallest phase is Anaphase. So in Anaphase, as chromosomes are already attached to the spindle fiber, so these spindle fiber, now the centromere of the chromosome, it divides into two parts due to contraction of the spindle fiber. So in Anaphase, centromere of the chromosome separate out. It divides into two parts, and this division takes place due to contraction of the spindle fiber. So spindle fiber contracts, and as these spindle fiber contract, they divide the centromere into two parts. As centromere divides, these chromatids, they get separate from each other, and as they separate from each other, they start moving towards the poles of the cell. The same change occur in plant cell also. Centromere divide due to contraction of the spindle fiber. Now, as centromere divide, it results in the separation of chromatids, that sister chromatids, and they start moving apart from each other towards the pole. At the end of the Anaphase, sister chromatids, they are moving. At the end of the Anaphase, these sister chromatids, they after separation, they are moving towards the poles, and they are nearer to the poles at the end of the Anaphase. Same in the plant cell also. Centromere attaching the two chromatids divides or split. Two sister chromatids of each chromosome separate and drawn apart from each other. They are pulled towards the opposite poles of the cell. Now, after this, one change which occur only in animal cell, not in plant cell. That change is that at the end of the Anaphase, furrows start in the cell membrane at the middle of the animal cell. Furrow formation begins in animal cell at the end of the Anaphase. Furrow formation takes place only in animal cell, not in plant cell. Why? Because in plant cell, cell wall is present, and cell wall is very rigid, so it do not allow cell to form furrow. So this furrow formation, it takes place only in animal cell, not in plant cell, and it begins at the end of the Anaphase in animal cell only, not in plant cell. So after Anaphase, the last phase of the cell cycle, that is telophase. In telophase, the two sets of chromosome reach opposite poles. So already during anaphase, as chromatids were moving towards the pole, so at the beginning of the telophase, they reach to the opposite poles of the cell. Now they had already moved to the opposite poles, so the spindle fiber disappear. Now their work is over, so spindle fiber disappear in telophase. Now the chromatids which had reached the opposite pole, there they decondense. They again decondense themselves to form long thread-like structures which are called as chromatin fiber. Now after this, the nuclear membrane and nucleus reappear. In prophase, this nuclear membrane and nucleus disappear. Now here, as the cell karyokinesis is going to over, so the nuclear membrane and nucleus, they reappear in telophase, or they are formed again in telophase. The furrow formation which already have started in anaphase, that furrow formation deepens in the animal cell. The cleavage furrow start deepening in the animal cell. So the furrow formation, this furrow, it becomes deep in the animal cell for cytokinesis to occur. Now in plant cell, furrow formation do not take place. Rest of the changes are same. Here also, chromatids on reaching towards at the end of the pole, they decondense and they again form long thread-like structures known as chromatin fiber. Nuclear membrane and nucleus is reformed here. After this, in plant cell plate formation take place, and the cell plate formation begins in telophase in plant cell. This cell plate formation will divide the cell into two daughter cell, that is, it will result in cytokinesis in daughter cell after the completion of telophase. Now, as the telophase is over, with end of the telophase, karyokinesis is over in mitosis. Means the division of nucleus is over, and after that, now the division of cytoplasm will take place, that is cytokinesis. And in animal cell, it takes place by deepening the furrow formation. So the cleavage furrow deepened totally in animal cell to split one parent cell into two daughter cell at the end of the mitosis, and that is cytokinesis. So by cytokinesis, one parent cell divides into two daughter cell. And in plant cell, this cytokinesis takes place by the cell plate formation which already had started in the telophase. So that cell plate formation divides the plant cells into two daughter cells having the same number of chromosomes. So this is all about 4 phases of the mitosis. Again, one revision. In prophase, the first change in animal cell, centrosome divide, centriole moves towards the pole. Spindle formation take place both in animal cell and in plant cell. In plant cell, spindle formation takes place by the condensation of microtubules, and here this spindle formation takes place in between two centrioles. Chromosomes are already duplicated, having two sister chromatids, one centromere, and at the end of the prophase, they begin to move toward the center, that is the imaginary center of the cell in both animal and plant cell. Nuclear membrane and nucleus both disappear in prophase. Now then, after prophase, cell enter the metaphase. In metaphase, chromosome reach the center of the cell, that is at the equator of the cell. They align themselves at the equator of the cell. And spindle fiber, they get attached to the centromere of the chromosomes in metaphase. Then in anaphase, these spindle fibers, they start contracting. As spindle fiber contract in animal and plant cell, they split the centromere that is the part of the chromosome. Due to splitting of centromere, chromatids also separate out, and with continuing contraction of the spindle fiber, these chromatids, they start moving towards the opposite poles of the cell. And at the end of the anaphase, still the chromatids are moving towards the pole, and in animal cell, in addition to it, furrow formation starts. But in plant cell, as rigid cell wall is present, no furrow formation takes place in plant cell. In place of it, cell plate formation will take place, but that will be in the next phase, that is telophase. So in telophase, already the chromatids have reached the pole where they decondense to form chromatin fiber. Nuclear membrane and nucleolus reappear. And furrow formation, the furrow which were already started, formation which has already started in anaphase, it deepens itself to divide the cell into two. In plant cell, the rest of the changes are same. Only the new change is that in plant cell, cell plate formation takes place to divide the parent cell into two daughter cells. And rest, chromatin fiber, they decondense themselves, reappearance of nuclear membrane and nucleolus. After that, cytokinesis, furrow deepens totally to divide one cell into two daughter cells. And in plant cell, cell plate formation has completed it during cytokinesis, which divide one cell into two daughter cells having the same number of chromosomes. So the difference between karyokinesis and cytokinesis is very important. Now after this, the differences in mitosis in animal and plant cell. The nuclear and chromosomal events of mitosis in plant cell are as the same as those in animal cell, but some differences are there. What are those differences? It is a very important question. Beta, in animal cell, esters are formed, radiating rays around the centrioles, but in plant cells, no esters are formed. Beta, in animal cells, cytokinesis occur by furrow formation, but in plant cell, cytokinesis occur by cell plate formation. Beta, mitosis in animal cell, it occur in almost all the body cells of animal, but mitosis in plant cell, it occurs at the cells which are present at growing roots and shoot, that is in meristematic cells only, divide by the process of mitosis. After this, now the significance of mitosis. Already you have done that significance of mitosis. Beta, the 5 significance of mitosis are: mitosis help the cell organism to grow. It help organism to repair damaged and wounded tissue by renewal of the lost cell. Beta, it also help in replacement of old and dead cells, such as the replacement of blood cells and the epidermal cells of the skin. It also help organism to reproduce asexually, that one cell divides into two daughter cells, that in lower organism asexual reproduction takes place by the means of mitosis. And the last, it helps the cell to maintain the same number of chromosomes in daughter cells. Next topic, that mitochondria and chloroplast in cell division. Beta, both mitochondria and chloroplast have their own DNA. As they have their own DNA, they also contain their own ribosomes. So they divide by their own by simple fission. They increase their size. As they have their DNA, they double their DNA quantity, and then they divide by the simple fission process, and they increase their number inside the cytoplasm of the cell. Now next is, how old are some of your body cells? Cells of the islands, nerve cells of the cerebral cortex, and most muscle cells last a lifetime, but once dead, they are not replaced. Skin cells are replaced every 2 weeks or so. But the red blood cells, you know that their lifespan is of 120 days and they are replaced after that. Bone cells are replaced every 10 years or so in adults. Epithelial cells lining the guts, gut means alimentary canal, last only for 5 days. And average life of other gut cell is about 15 years. And last, liver cells are replaced every 300 to 500 days. So with this, we have finished with the explanation of mitosis. Next, we will start with meiosis.